Yeah, today will be the week three. Unlike last time, there's nothing much to remember this time. I'll tell you some few concepts, and also we'll have a look at mostly at the Hardy-Weinberg equilibrium or Hardy-Weinberg principle. Actually, it is kind of tough. I think you may have doubt in that part, understanding Hardy-Weinberg principle. We'll also solve a question on that. So for today's session, I recommend you have a calculator with you. Phone calculator is also fine. If you're watching this Google Meet in your phone, then consider having a calculator with you. So we'll kind of solve a question also today. Okay then. Okay. So can anyone quickly confirm if my PowerPoint is visible to you all? Yes. Okay then. Great. Okay. So I'll start today's session then. Uh, Welcome to the week three problem solving session. I'm Rajesh. I'm a PMRF business scholar at the Department of Biotechnology, IIT Madras. And in this session, let's just so last time I was telling you, so in this session, we'll start uh, with a basic overview of the origin of life. So mostly I discussed this in very brief details in the first or the week one TA class. Uh, Let us discuss a little more about that into today's session. Then we will move towards the evolution, and then eventually we'll like ending with the Hardy-Weinberg principle. So like in the origin of life, what people had earlier idea or like some old ideas is that from the Greek time, like early Greek thinker, they thought that life units of life called spores were transferred to different planet, including Earth. So it came from outer space in the form of spore, which was completely absurd. And as scientists, we don't believe that. So that concept was called panspermia. So for a long time, it was also believed that life also like when people rejected this idea, the people thought that life on Earth it actually came out of decaying and rotting matter like straw, mud, etc. So that was the theory of spontaneous generation, which told that life generated out of nothing. So out of inanimate and inorganic matter, out of nowhere, life just generates like straw, mud, etc. But it was kind of disproved by these three scientists and their experiment. And this type of question you will see in MCQs that which three scientists disproved the theory of spontaneous generation. So those three are Francisco Redi, Lorenzo Spalzani, and Louis Pasteur. I think you are already familiar with these experiment, mostly this Francisco Redi or Redi-Jar experiment and Pasteur's Swiss Pasteur's experiment. And one more guy is also there, so all of these three people, their experiment is kind of more or less similar. So in Red Eye's jar experiment, he saw that when this piece of meat is open, the fruit flies or the flies are able to come and like lay eggs and maggots can generate on the flesh. But when it is sealed, then no maggots. Also, if the theory of spontaneous generation is true, then maggots would have originated from this decaying piece of meat out of nowhere. But since it is closed or it is not exposed to air or outside environment, so no growth is there or no life is originating by itself from a decaying matter. The same thing for the Spalzani experiment. he boiled media, and if it is kept open, then microbes grew. If it is sealed, or if it is not in contact with the extracellular, or sorry, external environment, then no growth. Similarly, Louis Pasteur did his famous swan neck that experiment. When he broke the swan neck, the media was able to come in contact with the outside air, and then this outside air actually has a different type of microbes, which then were able to grow in this broth. So by these three simple experiments, actually, these three people were able to disprove the theory of spontaneous generation, that life originates spontaneously out of decaying and rotting matter. Reddy expressed in his famous in his like theory, he gave a theory that omne vivum ex vivo, that all lives comes from pre-existing life only. So one life can only give rise to another life. For example, the outside environment. So the outside environment, which has microbes, only when they come in contact with a suitable environment, they can able to lay egg or can able to multiply, and then life originates from them. So something originates from the pre-existing life, or just to be simply said, all lives comes from life. So this was kind of very basic idea. After that, yeah, just one more. That's one question for you here. Try to answer this. The Reddice jar experiment challenges the idea of abiogenesis by what? Demo. You can put it in the chat box, or you can uh, unmute and speak. Yes, people. So, what did the Reddice jar experiment, like how the Reddice jar experiment challenged the idea of spontaneous generation? So it showed that life arises only in the presence of light, or providing that flies can grow in sealed environment, or showing that maggot can only develop from eggs laid by the flies, or demonstrates that microorganism can arise from broth spontaneously. So which one do you think is correct? Like people there, can you hear me? Or you need me to explain this answer? Okay, someone says option C. Others? Okay, I think I need to explain this one. Uh, okay, I think you are not familiar with the red eye star experiment, but all these things are already in your NPTEL module. So if you had seen the videos, you might have known this. Anyway, I'll explain you this. So the answer is option B. It shows that maggots can only develop from eggs laid by the flies. Thing is, okay, I'll go back and I'll show you this experiment, and I'll tell you about that experiment, red eyes jar experiment. So people told that life originated randomly or out of nowhere from decaying matter. So what he did is that he put a piece of meat here and let it decay. So once it decays, the flies will come and lay their eggs here, and from their eggs, the maggots will generate, and there will be a population of maggots. These are white color maggots. But if the same piece of meat is kept in a sealed flask, then flies cannot come inside and they cannot lay egg, and therefore no maggots. If this jar is covered with a gauze, like there is no direct contact, still the flies can come when the meat will start to rot, because air, it is in contact with air. So it will start to rot and the smell will go out. Attracted by the smell, the flies will come and lay their eggs here on the gauge itself, and then the maggots will develop here. So in the middle one, the flask, which is sealed, is kind of a control that it shows that when there is no contact with the outside environment, there is no maggots. So no flies, no maggots. So maggots is something new, some new life is originating. So it shows that if this piece of meat is not contact with the air, then there is no spontaneous generation of maggots. So for the maggots to germinate or arrive on the piece of meat, it has to be contacted with the outside environment. What is from where the maggots are coming? From flies. So simple statement is the maggots are not originating out of their own on the piece of meat. They are coming from the flies which are outside the jar. So that defines that life comes from life and not theory of spontaneous generation. There is no spontaneous generation. I hope you are clear now. Nandan, are you clear now? Nandan, are you clear now about this answer? Okay, I don't think he's there. Okay, fine. We'll go ahead. Okay. So that was about the red ice jar experiment. Okay, so as I was telling about a little more about the origin of life, uh, in the solar system of the Milky Way galaxy, Earth was supposed to have been formed. It is approximated that the Earth has been formed around 4.5 billion years back. So in a billion, there is 10 to the power 9 zeros. So at that time, when Earth was just forming, there was no atmosphere on the early Earth. So there's water vapor, carbon dioxide, methane, and ammonia. So note this term, methane and ammonia. They are released from the molten mass covered the surface. Uh, so the primitive Earth, there were a lot of volcanoes, and there was a lot of lightning strike, and the uh, environment was mostly methane and ammonia. So the UV rays from the sun broke the water. There were also water vapors. They water into hydrogen and oxygen, and the lighter hydrogen escaped. Oxygen combined with ammonia and methane to form water, CO2, and others. So likely then gradually the ozone layer was formed, and it is cooled the water vapor. So this water vapors, they actually cool down or got condensed and fell as rain to fill all the depression to form ocean. So it is also estimated that life appeared 500 millions year back after the formation of, life appeared 500 million after the formation of Earth, almost 4 billion years back. So kind of you can estimate it. If Earth is formed 4.5 billion years back, then life appeared post 500 million. So kind of 4 billion years back. So here the important part is that the atmosphere of Earth was methane and ammonia. So methane and ammonia both are reducing agents or they are reducing in nature. So thing is, the atmosphere of the Earth was reducing in nature. I hope I'm just brushing up what is a reducing agent. So reducing agent is a substance that is oxidized by donating electron or donating hydrogen. I hope you remember this from your chemistry concept. So example is here, like methane combining with oxygen, like NH3 here is getting an oxygen and giving out hydrogen to form water. So the agent which gets oxidized by donating electron or hydrogen is called a reducing agent. So methane and ammonia were in abundance, and this is how the atmosphere of primitive Earth or the early Earth was very reducing. So the modern theory of origin of life, they discard spontaneous generation. What they say is that the modern theory says that chemical evolution is the basis of origin of life. So first, chemical evolution occurred. So two scientists, Oparin of Russia and Halden of England, actually proposed that the first form of life could have come from pre-existing non-living organic molecule, that is RNA and protein, etc. And that formation of life was preceded by chemical evolution. So this organic molecule has to be formed from something. And these organic molecule actually formed from the inorganic constituents. So the condition of Earth, as I said, it was very high temperature, very high volcanic storms were going on, and it was reducing atmosphere, as I said in the previous slide, containing methane and ammonia. So to give an overview that how, or give an idea on an experimental basis that how organic molecule could be like produced or made from inorganic constituent. In 1953, Stanley Miller and Harold Urey created similar condition in the laboratory scale. In the right side, you can see this. So they created electric discharge in a closed flask which resembled the lightning during the primitive early Earth. The very high lightning, continuous lightning going on. And the atmosphere resembled the reducing atmosphere containing methane, hydrogen, and ammonia, and water vapor at 800 degrees Celsius. So 800 degrees Celsius, the continuous lightning is going on. And when that water cooled down, they observed presence of amino acid. And what amino acids they observed? Glycine, alanine, aspartic acid, nitrogen-based adenine, and simple sugar ribose. In addition, he found hydrogen cyanide and formaldehyde. So thing is, he observed or they observed the formation of amino acid, which is an organic molecule from simple inorganic constituents. So this is the possible or the overview of a reaction that would have occurred inside that led to the formation of the amino acid. This shows formation of glycine. It is already in your PPT shared by the NPTEL week. So it is just to give a brief overview that carbon dioxide splits into carbon monoxide and oxygen, and then methane can this methane can combine with oxygen to form CH2O, and similarly you can get. I'm just telling you the basic overview, brief overview. So this HCN you will get, and then formation of glycine can happen from the combining with CH2O plus HCN plus ammonia. See that it gives NH2 CH2CN. So this NH2 CH2CN can combine with a water molecule to give NH3 plus NH2 CH2COOH. So if you remember, what is the basic structure of amino acid? So it is NH2C. Uh, so to this carbon, there's one hydrogen and there's one COOH group. So there is COOH, there is NH2, there's one hydrogen, and R can be a variable group. So look here, this is NH2, C, one H here, CH2, and COOH. So this is a simple amino acid called as glycine. So I hope you can figure out that if you take inorganic constituent or inorganic gases in this a sealed environment and do electric discharge, you get amino acids. So that is the SL Miller experiment. Uh, I would suggest like all these reaction are not important. You did not memorize this. This is just for your understanding that how these gases can combine to form a amino acid. Just for your like knowledge, I put a structure of the amino acid, how amino acid look like. If you replace this R here with H, then it becomes glycine. I hope this concept is clear to everyone. Yes? Okay. Yes. Shall I move forward? Are you people following? Okay. Anyone? Just confirm. Like you people are following. I'll move forward. This is what I previously discussed. I'm just giving you a brief overview again. You people can unmute and speak or put it in the chat box.
Yes, sir.
Okay. Okay, great. Okay. So this is, as I said, we got organic molecules from inorganic molecules. And this concept is entirely in the PPT shared in that NPTEL. So you can, I think you must have gone through this. This is just a quick recap. So the according to the chemical theory of origin of life, there's a series of chemical synthesis which gave rise to life, what I just now discussed. So these large molecules, so this amino acid, uh, you can say these are kind of large molecules. So because amino acid can kind of link together and they can form a protein. So if you know the protein, the building block of protein is amino acid. So once we had a large organic molecule, it came together and kind of formed a colloidal aggregate. And the name, the like people call it is coacervates. From this coacervates, actually, the actual origin of life started by forming of first protocells and then the first cells. So before the first cell, there was something called as protocells. So what is not there in protocells? The enzyme, like the complex molecular mechanisms such as enzymes, were not there in the protocells. They had other ability, like to take up the new molecule to replace the degraded molecule and maintain their size. The coaggregates has the basic property of living system, but it does not have the complex molecules such as enzyme. Later, a nucleic acid is entrapped with coaggregates, and then process of division become precise and controlled. The this form of coacervates with nucleic acid is known as eubions or protocell. So protocell, remember, it is kind of like nucleic acid is there, but there is no enzyme. So enzyme is something which is advanced protein. So as there are just large organic molecule, there is still no new like protein, like advanced protein such as enzymes. So these are called as protocells. Then along with them, the in the next step or in the first cell, the protein molecule appearance of enzyme has enhanced the synthesis of several biomolecule in humans. Then RNA and DNA developed, and these from the nucleic acid, RNA and DNA developed, and these molecules have taken over the protein synthesis from the central dogma. The DNA from DNA, RNA arises, from RNA protein arises. So interaction of lipid and protein allowed the formation of biomembrane, which has provided selectivity in the primitive cell for intake and exclusion of material. So lipid membrane kind of formed, and these are very basic first cells. And of course, all these things were happening in water because this is uh, very much known that in the water only these large organic molecules form, and from uh, there the life originated in the water. So slowly this single cell organism became multicellular life form, and by the time of 500 million years ago invertebrates were formed and active. Then the jawless fish probably evolved around 350 million year, and then seaweed and few plant existed around 320 million years. Then gradually these reptiles came, and then gradually we had apes and then human. So all these things are just for your self-study. I'm just giving you a brief overview, and this is what uh, like people now accept as the modern theory of origin of life. Okay, so this is what the life originated. But how come from the origin of life we have right now the complex organisms? So one factor that led from simple organisms to the complex organisms that are present today is called as evolution. We will have a look at evolution like later, but solve these questions with the concept you just now learned. Okay. So the first one. The primitive Earth's environment is considered reducing because it has abundant of free oxygen, or it has low gravitational force or low atmospheric pressure, or it was rich in hydrogen and compound like methane and ammonia. Which one do you think? Okay. Yes. Anyone else? Yeah, okay. So this is the correct answer. As I said, the early Earth or the primitive Earth, the atmosphere was reducing. So it has lots of methane and ammonia. So the answer is option D. The next one. The modern theory of origin of life is distinguished from the earlier theories as it relies entirely on philosophical reasoning or based on experimental evidence given by Miller-Urey experiment, or it supports the theory of special creation, or it is a combination of religious and scientific ideas. Which one is correct? Yes. Yes, correct. Correct. Okay, correct. So, Miller and Urey's experiment proves that the modern theory of origin of life, which is chemical evolution. Okay, so it is option B. The organic molecule that are the precursor of DNA and RNA, according to the chemical theory, are amino acid like glycine and alanine, fatty acid like and glycerol, or polysaccharide like starch and protein, or sugar like ribose and nitrogenous bases like adenine. This is very simple question. What is DNA and RNA made up of? Yes. Oh, we have correct ribose sugar and yes, ribose sugar and the nitrogenous bases like adenine, guanine, cytosine, and thymine. And in RNA, it is uracil instead of T. Okay, so it is answer is option D. Okay, so as I said, like this evolution has happened. That means the advanced organism that are existing today, they had a primitive ancestor long, long back. and that is called something called as evolution. So what is the evidence that this evolution has occurred and they had a primitive ancestor? So the best evidence for evolution are the fossil. So fossil, what is fossil? That they are the remains of the hard part of the life form found in the rocks. So if you see the earth, there are layers of earth, or there are layers are called sediments. Different aged rock sediment can contain fossil of different life form who probably died during the formation of particular sediment. So if you go like deep into the sediment layer, you will find very old fossil than as compared to the younger fossil which are found in the top sediment. So it kind of says that different kind of organisms existed during a different period of time and they went extinct, and from them another set of organism or arised, and the organism that died are the ancestor of the today existing organisms. And all these evidence or all these fossil and this different layers sediments, they are called paleontological evidence, like the dinosaur bone and all. These are all paleontological evidence. Okay, so I told you this concept for this question. What is the direct evidence for the theory of evolution? Is it the DNA sequence, analogous structure, behavioral similarity between species, or the fossil record showing translational forms?
D.
Yes. Yes. Oh, it's D. So there are different evidence of evolution. Uh, the modern kind of thing or the more advanced is the DNA sequencing. Like there is similarity between different species of organism, and there is somewhat similarity between the unrelated species also, that came after the next gen sequencing and all. But before that, what is the hard and like the direct evidence what people can get is the fossil by digging the earth, by getting fossil from different sediments of the earth. So that is remember, if you see this type of question, that what is the direct evidence for the theory of evolution, it is fossil, because fossil show in different period of time different organism existed, and the fossil which are found in the older sediments are kind of linked or as look alike with the organism that are found in today's time. So they are kind of ancestor. So something might have happened, and this something is called evolution. We will have a look at this evolution and the forces of evolution. So this is option D. Fossil record showing translation form is the direct evidence of the theory of evolution. Another one, this one I have also discussed in week one. What is the method that provides an absolute age of fossil using radioactive decay? Is it morphological comparison, stratigraphic layering, or carbon dating or relative dating?
Carbon detecting.
Yes, carbon dating. Okay, so you people know it's carbon dating. Yes, so the answer is correct. Option C, carbon dating. Just to remind you, there's two type of dating. One is rubidium strontium dating, and one there is carbon dating. So there's two samples. Okay, one is rock, which is a inorganic sample, and another is organic sample. Like let's say this mosquito trapped in this quartz or in this stone. So whatever we will get from the mosquito is kind of organic matter. And let's say you want to identify the age of a rock. So that will be inorganic sample. So if you have inorganic sample, the method is called the rubidium strontium dating, which relies on the decay of rubidium to strontium, which is very, the half-life is very high. And by figuring out how much strontium 87 is there in the rock, you can relatively calculate. So it is very complex equation. I'm not telling you the equation, just the theory. By doing the rubidium strontium dating method, you can calculate the inorganic sample age. So as I said, if the sample is organic, then it is a radiocarbon dating or the carbon 14 dating. Remember, it is not carbon 12, it is carbon 14 dating. It is a scientific method that can accurately determine the age of organic material as old as approximately 60,000 years. Sorry. So what is this carbon 14? It is based on the fact that living organism can absorb carbon 14 into their tissue while they are alive. And when they die, this carbon 14 starts to change into other atom over time, or thing is that starts to degrade gradually. So scientists can estimate how long the organism has been dead by counting the remaining carbon 14 atoms. So during the lifespan of the organism, they will absorb the carbon 14 dating. As soon as they die, they will start losing this carbon 14. By calculating how much carbon 14 is remaining, you can kind of figure out that how long this organism has been dead. So that is called radiocarbon dating or carbon 14 dating. Okay. So before going to the more accepted theories of evolution, you people have one question about the embryological support of evolution. So this embryological support of for evolution was proposed by Ernst Haeckel. So this guy said that this actually the embryological support also for evolution is there because he observed that certain features during the embryonic stage common to all vertebrates that are absent in the adult. For example, embryo of all vertebrate, including human, develop a row of vestigial gill slit just behind the head, but it is functional organ only in the fish and not found in any other adult vertebrate. So if you see on the right side, there is gills in the embryo, which is kind of characteristic for the vertebrates. But these gills are only active in the fish, but not in the like this fish or in the human. We don't have gills. So Ernst Haeckel said that since this chicken, human embryo, and the fish, they have gills during the developing embryo, they are all linked and they have a common ancestor. So that is the embryological support for evolution. But you don't have to remember this because this theory was disapproved by Carl Ernst von Baer, as he noted that embryo never passed through the adult stage of other animals. Like human, like after certain weeks of development, the human embryo loses this gill or a tail. Tail becomes tailbone, and uh, the gills become the part of the head and neck region. So it was simply disapproved. So I'm just telling you this concept because you have one question from here. What is, in case you want to know, what is embryological support for evolution? There's a small video for you here also, which shows how simple cell originated or single cell originated, and then it gave rise to archaea, then eubacteria, then protozoa, and then more advanced organisms. So once you are done, you can later download this PPT and have a look at this. So the next, as I was telling, you have a one question from here. What is the main concept of Haeckel's biogenetic law? That embryo development reflects evolutionary history, or the evolution is guided solely by environment factor, or organism evolve by acquiring trait during their lifetime, or complex organism appear spontaneously. Which one do you think is Haeckel's biogenetic law? Yes, yes, correct. So Haeckel said that embryonic development reflect evolutionary history, which was later kind of rejected. So that was about Haeckel's embryonic law of evolution. So now I'll be coming, talking a little more about the more accepted theories of evolution. So there's something called as comparative anatomy and morphology that showed a similarity and differences among organisms of today and those existed years ago. So we will see how we have similarity and differences like to do the same function, and if this similarity or differences are there, then a common ancestor might be there in the past. So first we'll look about the similarity. So similarity can be interpreted to understand whether common ancestor were shared or not. So if there's something common here, like let's say there's common organism here, then different type of species that originated from this common ancestor would have similarity in at least some of the internal organ. If they do not even look alike, their internal composition or the skeleton, like the morphology of or the anatomy of the bone will be kind of similar. And this is what we observe. Is anyone telling anything? Yeah. The- Sevas, do you want to ask anything? Okay. So I'll finish this. So if you consider different mammals, for example, whale, bat, cheetah, human, or in the picture you can see this human hand, cat hand, horse hand, bat hand, and dolphin hand. Dolphin or whale, they both belong to the mammal. You can see, although these animals do not look like each other, but the forelimb perform the different function in all these animal, but they share the similarity in the pattern of bone of the forelimb. So what similarity is there? They have similar anatomical structure. All of them have humerus, radius, ulna, carpal, metacarpal, and phalanges. If you look clearly, their shape might be different. Their function is uh, kind of different. Like human use hand for different purpose, cat use their forelimb for different purpose, horse for different, bat for flying, dolphin for flipping in the water, mostly different function, but the internal anatomy is similar because they all have humerus, radius, ulna, carpal, metacarpal, and phalanges. And this label of similarity may not come if they do not have a same ancestor, or if they would not have originated from a same ancestor or organism which existed long back. Hence, in this animal, same structure developed along different direction due to adaptation to different need. So based on their need, dolphin to like move in the water and back to fly, they developed the same structure got developed along different direction due to adaptation to different need. And this is called divergent evolution. And the structure alcohol called homologous indicates homology indicates common ancestry. So this kind of rays coming out of the common ancestor, and that is why it is called divergent evolution. So from one, it is coming out to many, and that is why it is called divergent. So it is diverging. The rays are diverging from the common ancestor, and since they have all radius ulna carpal metacarpal, they are called homologous organs. So remember, we have homologous organ in the case of divergent evolution. Now let us look at something uh, different or something upside down. So it that was homologous organs. They have different function, but they look alike. In the case of analogous organ, they have same function, but they look different. For example, the bat wing, the bird wing, and the insect wing. The bat wing, they have a different bone architecture than the bird wing, and the insect wing do not have the solid bones and all. There's a smooth wings in case of insect, but all of them perform the same function, that is flying. So what I was trying to say is that the wings of butterfly and the birds look alike. They are not anatomically similar in structure, though they perform the similar function, that is flying. Hence, analogous structure are result of convergent evolution. So for bat, for bird, and for the insect, there might be different uh, ancestor, but based on their need, that is to fly, they originated uh, similar kind of organ. That is why it is called analogous organ. So remember this, they are different structures evolving for the same function and hence having similarity. Since they are converging to one point, it is called analogous organ or convergent evolution. In the previous one, it is originating from a single ancestor and radiating based on the need. That is why it is called divergent evolution. Here it is originating from the different ancestor, but coming together or converging together to perform the same function. That is why it is called convergent evolution. There it since it is originating from single ancestor and going toward making performing different function, it is called homologous organs, and these are called doing the same function, it is called analogous organs. So two things you have to remember. Homologous organ we see in case of divergent evolution. You don't have to remember this like it or make this very complex and remember. Just remember divergent means originating from a single ancestor going in different direction due to different need. Since there is different need, they are, they might look different. The structure of the organ as they have different need, they are performing different function. But since they originated from a same ancestor, they have same kind of internal anatomy. That is why homologous. Since in the case of convergent evolution, they are coming from different ancestor, They have different type of organs. That is why they are called analogous organ, and the name is called convergent evolution, converging to do the same function. We can have also other example of analogous organ. Example is the eye of an octopus and of a mammal, or the flipper of penguin and dolphin, kind of doing the same function, but they are anatomically not similar. We can also see this thing, the convergent evolution in the case of plants also, like sweet, the normal potato is a stem modification, but in the case of sweet potato, it is a root modification. Both doing the same functions to a storage of starch. And these are the example of analogy. Okay, I hope this concept is clear to you all. Homologous organ and analogous organ. Homologous is associated with divergent evolution. Analogous organ is associated with convergent evolution. Okay. Any questions? Anyone would like to ask anything, or shall I proceed? Okay. Okay. If not, we'll go to the questions. Some of the question just now discussed, the concept or based on just now discussed concept. So the principle that underpins the concept of homologous organ is what? Spontaneous generation, parallel evolution, convergent evolution, or divergent evolution. Homologous organ.
See,
Yes. Okay. So you people are following. Okay, good. So homologous organs are associated with divergent evolution.
these diversities.
Yes. Since they originated from a same ancestor, they have homologous organ. So that, but they diverged to accommodate different function. Okay, so option D. Okay, now we have evolution, but what is the force that drives evolution? From one ancestor, what is the force that drove that ancestor to make different kind of other type of species, which is some more or less related to the ancestor. So one force is a natural selection. So this natural selection, it was given by Darwin. So Darwin said that who are better fit in an environment leave more progeny than others. And these therefore will survive more and hence are selected by the nature. Darwin called this natural selection and implied it as a mechanism of evolution. So the force of evolution or the mechanism of evolution, they are same. One is natural selection, which I am talking about it now. Another one I'll talk about later is mutation. So natural selection, that all existing life forms share similarity and share a common ancestor, but some survive more due to because they are better fit in the environment. One evidence or one example that supports natural selection is that the observation that comes from England. So there were moths growing in the tree, and people saw that the collection of moths made in 1850, that is before industrialization, it was observed that there were more white-winged moths because before industrialization there was not much smoke and the dust particle or the black smoke coming out from the industry. So the tree on which the moth reside is kind of the bark of the tree is white, so that favored the white winged moth, and the black colored moths are generally contrasting in nature against the bark, and they are easily picked up by the predators. But a collection made after the industrialization, that is in during the 1920, people saw there are more dark-winged moths. So what happened, like after industrialization, there is lot of pollution or black smoke coming out, and the trees nearby became black in color. Now the moths which were black in color had the ability to camouflage in the background, and the white colored moth, which was kind of contrasting in color, got picked up by the predators. So that is why the black winged moth, post industrialization, their population grew in number and their population declined in number. So this is called something called as natural selection. And this is one example of natural selection. So what is the other mechanism? I'll just give me a second. Just a second. Mm. Hmm. So that is natural selection, which was like this concept was proposed by Darwin. The another concept is called mutation. So what mutation into the picture is Hugo Debris. Hugo Debris, he was working on the primrose, evening primrose, and brought forth the idea of mutation. So mutation, what is mutation? That it is large difference arising suddenly in a population and mostly reproducing sexually. He believed that it is mutation which causes evolution and not the minor variation that Darwin talked about. But now we know that it is a combination of mutation as well as natural selection. Mutation are random and directionless, while Darwin variation are small and directional. Evolution for Darwin was gradual, while Debris believed that mutation caused a speciation and hence called it saltation. So Hugo Debris said mutation is a sudden change, uh, and this is the cause of evolution. And this he termed as the term he gave was saltation, single step large mutation. So we will see that during in the next week we will be looking at meiosis. So in meiosis we will see there is crossing over, and this results in the variation. So um, this is the two concept of evolution. One is slow and the steady, which is given by Darwin, which is called natural selection, and which is more frequent or large changes, which is called mutation. And mutation, much more responsible, the thing which is much more responsible for mutation is sexual reproduction. Like in this picture, you can see in the same plant, if you cross these two white flowers, out of nowhere, a pink color flower originated. So something, some nucleotide base in the gene sequence might have changed due to mutation, which led to this sudden change or such a big change that the color of the flower changed. And this is the work of Hugo de Vries on the evening primrose. Okay. So answer this question based on these concept. The role that mutation plays in the evolution is what? Ensures all offspring are identical, generate genetic diversity necessary for evolution, eliminate harmful alleles, or it causes uniformity in the genetic makeup of the population. Okay. So this mutation concept, mutation and variance is with applicable for us also. Like there is some mutation between while in the formation of gametes and all. That is the reason why no two individual human being look alike. We look different from the other human because there is some changes in the nucleotide base that is nothing but called as mutation, and this mutation results in the variation. So now you can think that what role the mutation plays. Yes, someone said B. Yes, so correct. So the answer is option B. Mutation generate diversity necessary for evolution. So what I said in the previous slide, that mutation, there are two forces for the evolution. One is natural selection, one is mutation. Mutation is the large single step, large number of this one single step, large mutation, which Hugo de Vries believed that mutation can cause evolution much faster. Okay. So the sexual reproduction is considered as advantageous for evolution as it ensures clones are produced, it reduces mutation rate in offspring, or it introduces genetic variability through recombination, or it allows for faster reproduction compared to asexual reproduction. Which one do you think sexual reproduction is advantageous for evolution?
C.
C. Okay. Yes. So sexual reproduction is the fusion of gametes. Gametes are generated by meiosis. In meiosis case, when the crossing over happen, there is like first of all, you will get the different mutations in the like not very large mutations, some minor mutations, but these minor mutations are enough to create a genetic difference or genetic variability. Okay, this is option C. Okay, people. Now, please consider getting your calculator. So there's nothing more to memorize things. Now we will uh, have some application-based question. And I don't know if you think Hardy-Weinberg principle is very difficult concept, but after this you will feel it is a very easy concept. Okay. So before going to the Hardy-Weinberg principle, just let me clear this concept allele. I don't know if you people know what is allele or not. So let's say you have a characteristic or a phenotype, let's say tall and short. So you all know we are diploid organisms. We have two sets of chromosomes. For any feature to express or for any phenotype, there is particular genotype. So for in this case, let's say the
Define
genotype.
the allele, sir.
Sorry.
Allele. Define allele.
Yeah, that's what I'm telling you. That's what I'm telling you right now. What is allele? I just before going to the Hardy-Weinberg principle, I'm just clarifying this concept. So these will be useful in the next slide. So I don't know if you people know what is a allele or not. So I'm just telling you what is a allele. So if you remember this capital T small t, crossing of the capital T small t, which resulted in the tall plants. Do you know this? The capital T is a dominant allele that resulted in the tall plants. Okay, so forget this. So let's say a human being, this is a tall human being, this is a short human being, the height of the human being. And let's say the gene responsible for the height is defined as capital A, for example. And as you all know, we are diploid organism. We are diploid organism means we have two sets of chromosome. We have a copy or a double copy of chromosome. So for this tall or short phenotype, there would be different some genotype also. And it cannot be capital A, it cannot be just small a because we were diploid organism. It has to be double. Either it has to be capital A, there has to be something present in the other chromosome also. So if it is capital A, it is tall. Because let's say capital A is able to dominate small a. If there is too small a, then that is short. If there is capital A small a, then it is still tall because capital A is able to dominate small a. So what is this capital A? What is the small a? It is the occurrence of a gene in the chromosome. So as I said, gene A is responsible for the height. What is gene? Gene is a part of the DNA. So there will be gene A in somewhere in the chromosome. Since we are diploid organism, there has to be something on the other side of the chromosome or in the chromosome 2, like this one. So we can say for a gene, in this case A, there are two alleles. Alleles, what is allele? Allele is the different form of the gene. Either it can be capital A or it can be small a. I hope you are clear now what is allele. So allele is the different forms of a gene that are occurring in the chromosome, different in the two sets of chromosomes. If capital A is there, small a will be there, then it is a heterozygous condition. If capital A is there or small a is there, then it is, sorry, if capital A is there, capital A is there, then it is homozygous condition. Small a is there, small a is there, then it is also a homozygous condition. Okay, people. So I hope you are now clear. See, we have homozygous condition if we have both capital A, heterozygous if we have capital A, small a here, or small a here, capital A here, and homozygous condition if we have small a, small a. So two concepts are there. Homozygous if we have similar allele of a gene. Heterozygous if we have different allele of a same gene. So allele is a different form of a gene which is present in the chromosome. Now everyone just confirm that you are clear with this much concept. Then we will further proceed. Uh, You are clear with the allele, you are clear with homozygous, you are clear with what is heterozygous. Very simple to understand, nothing more difficult. This is like a continuation from the Mendelian theory. Everyone is clear what is allele and what is homozygous and what is heterozygous condition. Anyone? You can put in the chat box also. This is important to understand Hardy-Weinberg principle.
Yes, sir.
Okay. Okay. In case you are wondering why, how come there's two sets of chromosomes? So we have sexual reproduction, a sperm fusing with egg. So sperm will have one set of chromosome, egg will have one set of chromosome, and now they are fusing randomly. This chance event. You can get capital A, capital A, capital A, small a, capital A, small a, small a. That is completely based on the random fusion of egg and sperm, which generates different uh, conditions such as homozygous or heterozygous and different combination of the allele. Okay, so I'll go to the Hardy-Weinberg principle then. So Hardy-Weinberg principle states that in a given population, one can find out the frequency of occurrence of allele of a gene or a locus. So now you are clear about allele, capital A, small a are allele of the gene height, let's say height. So Hardy-Weinberg principle can find out what is the frequency of the allele. What is the frequency of capital A? What is the frequency of small a? What is the frequency of capital A, small a? So what do you mean frequency? Frequency is a simple term. How many times it is occurring? If there are, let's say, 100 event, how many time this event is happening? So that is called frequency. Simple English term, frequency. So Hardy-Weinberg principle can figure out the frequency of occurrence of the allele. And this frequency, they said, is supposed to remain fixed and even remain the same throughout the generation. Hardy-Weinberg principle stated it using the algebraic equation. We'll come to the equation shortly. If I'll tell you the equation, it won't make sense now. So according to them, the gene pool or the total gene and their allele in a population remains constant. And this is called a genetic equilibrium. And if you sum all the allele, it becomes one. Like uh, they're different alleles, right? But if you so to visualize what I'm talking, let's imagine 100 population. There are 100 people. They will have different allele, but the population size is 100. So if you consider all the difference of the allele, and if you add these, the total has to be 1. So it is mathematical unity. Like if you are considering something and dividing into parts, or if you are considering one whole and you are dividing into a small part, if you put together that small thing, it will become one. It is not something which will exceed one. So it is kind of theory of unity. So that is what Hardy-Weinberg principle is. That you can figure out in a population what is the frequency of allele, and in a population if you add this frequency of allele it will become 1, because this population is not changing, it is a constant population. And this is one assumption of Hardy-Weinberg principle, that the population size is constant. This I'll tell you later. Okay, so I hope only the definition part, only the definition of allele and the definition of Hardy-Weinberg principle is clear. I'll explain you in detail in the next slide. Just tell me this definition is clear. Now you understand what is the definition of Hardy-Weinberg principle. Yes, you can put in the chat box also. Now we know what is allele, what is frequency, what is Hardy-Weinberg principle definition. Yes, is it clear?
Yes, sir.
Okay. Uh, Nothing much like a very difficult concept here. Okay. So keep it aside. I was telling you about allele frequency. So simple English word frequency, how many time this event is occurring in the total number of event. Let's say event is 100. This event is happening 7 times. So the frequency is 7 divided by 100. Now coming to the biological concept, applying the same logic, tell me what is the allele frequency of capital W and small w. So on the left of your picture, we have different type of flower in the Mendelian experiment as you saw. Capital W, capital W result in the violet flower. Small w result in the white flower because capital W is completely dominant over small w. So we will have different types of like homozygous or heterozygous condition. We might have WW or capital W small w or small w small w. Now consider there are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. There are total 9 flowers with their allele condition. Now you take this question and tell me what is the frequency of capital W and what is the frequency of small w. Just tell me or calculate yourself before me telling you this answer. First, tell me what is the frequency of capital W. I'll just give you a very simple hint. Just calculate how many times that capital W is there in the total allele. So capital W is allele, small w is allele. Just calculate how many times this capital W is there in the total allele. So that will be allele frequency. Simple mathematics. Okay. Anyone able to solve? What is the allele frequency of capital W? Yeah, you can try. Just tell me if it's fine if you're wrong. Then only you can understand what does this allele frequency mean and how to calculate the allele frequency. It's like calculating your percentage. So how much you scored divided by the total score. People solve this very simple. Anyone able to get the allele frequency of capital W? Okay. Okay, then listen here. So frequency, as I said, frequency is something how many time.
3 raised to 3, 6 raised to 3,
Sorry. Can you just tell me in the decimal? Just divide it and tell me in the decimal number. Like
6 raised to 3,
0.6 is to No, I'm asking frequency. So frequency is something dash divided by dash. Okay, okay, listen here now. Otherwise it will be very late. So I'm asking what is the just can you just mute your microphone? Okay, so I'm asking what is the frequency of capital W and small w. So frequency is how many times capital W is present in the whole. So how many times it is like what is the first you have to calculate that what is the total allele and how many times W capital W is present or small w is present. So first question, how many total allele is there? One, two, simply you can calculate. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Simple. So we have total, we have 18. Out of which, how many times capital W is present? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, divided 13. So frequency of capital W is 13 divided by 18. It comes out to be 0.72. Simple. That's what I'm asking. What is the allele frequency of capital W? Now tell me what is the allele frequency of small w?
0.27, sir.
Sorry, point two, point how much? I couldn't hear you actually. You can put it in the chat box also. There's nothing to be afraid of. You can freely speak and you can freely put in the chat box. It's okay. So small w is how many time? 1, 2, 3, 4, 5. 5, sorry, divided by total is 18. 5 divided by 18. Yes. So it is coming out to be 0.28. You can say the full 8, no? 5 divided by 18. Is my calculator working wrong or what? 5 divided by 18. Yeah, it is 0.2777. If you approximate, it is coming to be 0.28 in two decimal numbers. You can write 2.777, but you are correct. It is 277, it becomes 0.28. 0.28, 0.72. Now observe here. If you add this and if you add this, how much it is coming? Total is 1. So that is the Hardy-Weinberg principle. If you add the allele frequency, it comes to 1. All clear? Quickly tell me. Everyone is clear about this?
Yes.
Okay. So most of the Hardy-Weinberg principle is done. Now you calculated capital W, you calculated small w. Let's just give them some number, some word. The allele frequency of the dominant or the dominant allele here, the capital W is dominant over small w because even if it is capital W, small w is there, it is still violet flower. So that means this is capital W is dominant. So we'll give some random, not random, we'll give another word, sorry, another letter for these. So for the allele frequency of the dominant allele, we will give this letter P For the allele frequency of the recessive or the not non-dominant allele, we will give this letter Q, P and Q. Here I have written P and Q. So this part is clear. We calculated the allele frequency of the dominant allele W, capital W, and the allele frequency of small w, of allele frequency of the recessive allele, small w, and we assigned them P and Q respectively. Now let's move forward. And here we calculated that the allele frequency of small p is 0.72 and the allele frequency of small q is 0.28. Allele frequency of P and Q as 0.72 and 0.28, and when we added them it became 1. And this is most of the Hardy-Weinberg principle. Now, if you remember the uh, definition here, where is that? That Hardy-Weinberg principle defines this frequency of occurrence of allele in terms of algebraic equation. Not very difficult equation. I'll show you how we can express this in terms of algebraic equation by this example. So we saw capital W, small w, and we found out the frequency. Let's say uh, here it is given that the frequency of capital W, so in simple word, allele capital W and small w are present in egg and sperm in the proportion to their frequency in the population. This is the given in the question. That it is given that the frequency of capital W is 0.6 and the small w is 0.4. Then we can calculate the expected genotype frequency. What will be the different genotype frequency? So for the capital W to produce a capital W, then the egg and sperm must be having capital W. So the probability of producing a capital W organism is 0.6 multiplied by 0.6 because the allele frequency of capital W is 0.6. So it is 0.36. That is the probability of getting a homozygous capital W. Similarly, we can calculate what is the frequency of getting a homozygous recessive small w. So 0.4 multiplied by 0.4, since the small w allele frequency is 0.4, it is 0.16. Now the question arises, what is the allele frequency of the heterozygous condition, capital W, small w? So either in the sperm there can be capital W, in the ovum there can be small w, or in the sperm there can be small w, and in the ovum there can be capital W. So now you can understand what I'm trying to tell. So since before going to that, since we just multiplied this like 0.6, 0.6, so it was initially P, now it became P square, and initially it was Q. So we multiplied these two, now it became Q square. Now how to calculate this heterozygous condition, capital W, small w? That is what I just told you, that the sperm contain capital W or the egg has small w, then just multiplies these two, 0.6 and 0.4, or the reverse. Egg contains capital W, sperm contains capital small w. So it is again 0.6 multiplied by 0.4, 0.24, 0.24, 0.24 twice, because this can happen twice. If you are not clear, see this picture. Capital A, small a can be here, small a and capital A can be here. Both is called as heterozygous capital A, small a. So in heterozygous condition, it is 2 multiplied by the allele frequency because there is two chances. Here The egg has capital A, sperm has capital A. Here the sperm has small a, here the like egg has capital A. There's no other possibility. Here there are two possibilities. That is why we are multiplying with 2. Makes sense, right? Logical. So the probability of capital W and small w is 0.24 plus 0.24. What we got here is 0.48. So that is what is the genotype frequency in the next generation. From this, you can, if the allele frequency is given, you can calculate what is the genotype frequency of the next generation by using Hardy-Weinberg principle. Note one interesting formula here. Here, capital W got multiplied and become P square. Here, small w got multiplied and become Q square is Q square. Here it is P multiplied by Q, PQ, and here also it is P multiplied by Q, PQ. And here it becomes twice, so 2PQ. So P square plus Q square plus 2PQ is what equation? P plus Q whole square. And if you also observe, in case you didn't observe this, it is not working. If you add this 0.36, 0.48, 0.16, it all becomes 1. So in the next, I'll bring the equation now into the picture. Since you are clear with allele frequency, what is P, what is Q, how come we got P square, how come we got Q square, and how come we got 2PQ. So this is the formula for the Hardy-Weinberg. So it says P square plus 2PQ and plus Q square is equals to 1. P square is the homozygous dominant, Q square is the homozygous recessive, and 2PQ is the heterozygous condition. Twice because this can go both way. And if you add this, it becomes 1, where P, small p, is the frequency of allele, dominant allele, Q is the frequency of recessive allele, and in our example, we got P as it was given here, P as 0.6 and Q as 0.4. So here it is 0.26 square plus 2 into 0.6 into 0.4 plus 0.44 whole square. It is 1. So this is the general formula for the Hardy-Weinberg equilibrium or the equation. Okay, I hope till this part everyone is clear. Don't get confused like PQ, PQ. But while I'm telling you something, just keep on writing one line, and then you will figure it. This is not very difficult. I just started from the starting. What is allele? What is allele frequency? How we denoted P and Q, and then if there is twice, you have to multiply it twice. If you multiply P twice, it becomes P square. If you multiply Q twice, it becomes Q square. If you multiply PQ, it becomes PQ. If you multiply, but there's two conditions or two possibility of PQ, so 2PQ. And if you add all the allele frequencies, it will be 1. So this is the Hardy-Weinberg principle. Okay. So I hope this till this part it is clear. I have one question for you. Then you can be more clear about this Hardy-Weinberg principle. Yeah, people, you are following. Till this, it is clear how this equation came into picture. Anyone? Don't worry. You can download this, go through the slide by yourself. If you're still not following, we'll discuss it in the next class also. in the starting 5-10 minutes. But till now, are you able to like get a hold of this Hardy-Weinberg principle? Yeah, people. So I'm running out of time. I just have 20 more minutes. And I have one more, one simple question, not a long, like a very long question, one small question based on the concept. Just don't have to do anything. You just have to remember what is the allele frequency. In most of the question, allele frequency will be given and something will be asked. You just have to figure out what is P, Q. P is the dominant allele frequency, Q is the recessive allele frequency. That's all. These two. I hope all are clear. We'll go to the next slide. Yes, come on. Confirm anyone. Are you following? Yeah, okay. So then I'll go to the next slide then. But please download this and solve it by yourself. I'm telling you, this is very simple. This is not at all difficult. It looks like it is difficult. It is not. Okay. But thing is, why we need to study all this P, Q, P square, Q square? Does it has any application? So if you solve this question or if you go through this question, you can figure out what is the application of this Hardy-Weinberg equation. So here the question is given that 9 out of 100 individual in a population suffer from homozygous recessive disorder like sickle cell anemia, cystic fibrosis. Here for you, the question is, can you calculate the frequency of the disease causing allele? So here the disease causing allele is a homozygous recessive disorder. I hope can already figure out what I am talking about. What is P or Q is given here. 9 out of 100 individual homozygous recessive disorder. Question is calculate the frequency of disease causing allele. Okay, someone I think put in the chat box. Okay, 0.09. Okay. Okay, let others also solve this. Then we'll go to the second half of the question. It's okay, you can try and you can write in the chat box. Even if it is wrong, doesn't matter. You will get the idea. 9 out of 100 individual homozygous recessive disorder. Calculate the frequency of disease causing allele. Uh, I'll just tell you again. P is the dominant allele. Q is the frequency. P is the frequency of dominant allele. Q is the frequency of recessive allele. Here it is asking for the frequency of disease causing allele, which is a recessive. Anyone else? Uh, okay. The, the person who said 0.09, uh, just have a look at here. Homozygous recessive. So the frequency is 9 out of 100 and it says homozygous recessive. So there is 2. That's how this word called homozygous also. Okay, I won't take much time. So see this question, how this is solved. So it says it is 9 out of 100 individual homozygous recessive disorder. So for the homozygous condition, there has to be, like in the previous case, there is small w, small w. Here, let's say it is small a, or depends on what you want to give the name of the gene, disease causing gene. Let's say small d, small d. So the frequency is 9 out of 100. So the frequency is 0.09 of the individual are homozygous recessive allele. And it is q square, not q, because it is, as I said in the previous slide, it is human, we are diploid organism. So the frequency has to be there in the form of twice. So it is 0.09 is twice. So it is q square. I'll just quickly go back in this one. See here. The homozygous condition, it is small w is twice and it is q square because we are giving q as the frequency of a single allele, that is the recessive allele. So for the recessive allele homozygous condition, the small a has to be twice, so it is q square actually. Now you can answer. So q square is actually 0.09. Then you can find what is q, which is 0.3, which is the frequency of allele small. Yes, correct. So here it has asked the frequency of the disease causing allele. So here the disease causing allele is, so let's say small a, not the capital, because it is a homozygous recessive disorder. Homozygous recessive disorder means the recessive allele has to be present twice, small a, small a. So small a population is the frequency, the small a population is 9 out of 100. So the small a means q twice. So that is q square. Q square is equals to 0.09. But here you are asked to find out what is q. So from q square you can find out q. Now I'll go to the other part of the question. Can you calculate how many heterozygotes are there in the population? Heterozygotes means capital A, small a. And one hint is already given. Because P plus Q is equals to 1, as I said, it has to be 1. Okay, sorry. Okay, 16. Okay, others, can you try? Okay, heterozygotes are 16. Okay, can others also try? Now you found out the allele frequency of allele frequency of the recessive allele, that is Q, 0.3. You already know that P plus Q is equals to 1 because it has to be 1. So you found out P is equals to 1 minus 0.3, it becomes 0.7. Now you just have to plug P and Q into the equation. In the previous slide, I showed you the equation. Just solve this and we'll stop it here today. Enough of questions for today. Okay, so 16. Okay, okay, I won't take much time. So here you found out the value of P by subtracting 0.3 from 1 because P plus Q is 1 and Q you found out 0.3. Now you have with you have Q and you have P. 42. Okay, yes. So you find out what is P square. It is 0.89 and this is the frequency of homozygous capital A, capital A. But the question asked here is that can you find out the heterozygous? So 2PQ. Now you have P, you have Q. You just have to multiply. 2 multiplied by the frequency 0.3 multiplied by 0.7, it comes out to be 0.42. Given the population is 100, so if the frequency is 0.82, how many individual? Just multiply the frequency with the total size 0.42 multiplied by 100, it became 42. So the answer is 42. Okay. I hope you people understood, but I also know that in just one go, this might sound very difficult. I try to make it as easy as possible, but I would suggest that I'll upload this. I have couple of more slides about Hardy-Weinberg principle. Like this is done. This is the Hardy-Weinberg principle is over. What is the condition of assumption and what is the like condition of Hardy-Weinberg principle? How it indicates evolution? This I'll tell you later. Enough of a very heavy task today. I'll suggest you download this PPT. You take a piece of paper and start writing from here. Understand by yourself what is allele, what is homozygous, what is heterozygous, what is frequency. Once you understand this, then calculate the frequency yourself. Once you calculate the frequency of the dominant allele, give them P. Once you calculate the allele frequency of recessive allele, give them small q. Then you go to this page and here allele frequency are already given. This is different question. Allele frequency is already given. You are asked to calculate the genotype frequency. Then you multiply the P and Q and then C because capital W is the homozygous dominant genotype. So this will be P multiplied by P is P square and Q square. Then you see after you get P square, Q square and your 2PQ, add these and then get 1. Then you can figure out what is this equation and then you can try to solve this question by yourself. Then you are done understanding Hardy-Weinberg equation. Okay, so I'll stop here for today.